Absolute pleasure to be here, and thank you so much, Nancy, to the organisers for inviting us today. To talk about the work that we do at Healx, which really is, as it says on the title of the slide, all about accelerating treatments for rare disease patients. What I'm going to do today, kind of over the next 30, 40 minutes or so, give you a bit of an introduction to the Healx technology platform, talk a little bit about the artificial intelligence models that we've built enable us to accelerate treatment discovery for rare diseases and aiming to have an increased probability of success. Talk a little bit about the platform, but then I thought I'd also give a little bit of background story, so where Healx came from, what really was it that inspired Tim Gilliams, our CEO, and Dave Brown, our co-founder, to actually set up the company. I'll talk a little bit about the clinical trial development we're going through with our lead program, a bit of a case study around Fragile X. And hopefully what I'll be able to do in so doing is give you a sense that we are aspiring to achieve the mission that we've set out for the company. And as it says on the slide, very much our mission is to achieve unprecedented impact for rare disease patients by pioneering the next generation of drug discovery. So what was it that inspired Tim Gilliams and Dave Brown to found Healx? And it really is this chap over here pictured in the top right image. This is Nick Saru. About 8 years or so ago, Nick was giving a presentation at a patient group organised symposium. Tim and Dave very much in the audience. Nick is a parent of 2 children with a rare disease, alkaptonuria, also known as black bone disease. And Nick was recounting his story of how he was trying to repurpose the active agent of a herbicide, a weed killer, nitrosamine. He couldn't understand that nitrosamine operated in similar biosynthetic pathways in plants as it did, as it potentially could have therapeutic benefit in humans. And he'd gone on a mission to repurpose this agent. Now, Nick, not a scientist, but managed to bring together scientists, clinicians, and funding to run a drug discovery and development program, repurposing a treatment ultimately to improve the quality of life of his own kids. What's really remarkable about this study is that Nick, in about 5 years' time, for fewer than around about $10 million, received EMA approval for nitrosamine as a treatment for alkaptonuria. That was approved last year. I should just say that the clinician helping the development of that clinical program, Tony Hall, is our Chief Medical Officer at Healx as well, very much part of that process. Now, Tim and Dave in the audience listening to Nick's story wondered, could we learn from Nick's experience? Could we apply, for example, artificial intelligence, big data, and not just develop a treatment for one ultra-rare disease, but start to address the medical unmet need in the larger rare disease community? There are, effectively over 7,000 rare diseases, 95% of which don't have an approved therapy. Put that into context, that's 400 million patients affected worldwide, which is effectively larger than the population of North America. Clearly an area of large medical unmet need, and one that Healx was established to try and address. So how is it that we're going about doing this? What's unique and disruptive in Healx drug discovery? And as it says at the top of this slide down here, what we're aiming to do is apply algorithms, big data to large sources of varied inputs to find connections between the drug and the disease. Ultimately, in what we're referring to as a hypothesis-free or target agnostic approach, trying to limit the impact of human bias in trying to find novel treatments for rare disease, ultimately with the intention of either producing monotherapies, single-agent drugs that could be ultimately directly repurposed, then if you came to Nick's story, or indeed, you'll see in a moment that we have algorithms that predict synergistic combinations, potentially identifying pairs of drugs that might have greater therapeutic benefit in complex rare diseases than where ultimately one drug might just not cut it. We've also recently developing digital chemistry approaches that enable us to make small substructure changes to chemical structure, because In many cases, when we're repurposing drugs, it might be that in the new indication, the drug might potentially have off-target effects. It could give some aspect of toxicity, and we're trying to address that by making the compound. So effectively, what Healx is doing, which is disruptive in drug discovery, is allowing data and algorithms to find connections between a drug and a disease, which we immediately move into preclinical testing. And once we've detected those active drug candidates, we then step back to an understanding of what is the new biology or indeed the new mechanism in that new indication. Healx is a approach then. So let's start and have a look at the technology platform that we've developed in order to enable us to do this in a semi-automated way at scale, always thinking about that increased probability of success. So, fairly complex slide down here, but I will talk you through it. At the top of the slide here are the 4 key steps to Healx's approach to drug discovery. And of course, it all starts with data. What we're doing is ingesting large amounts of clinical, biological, and pharmacological information into a comprehensive rare disease knowledge graph, and that knowledge graph forms the input to our algorithms that solve the key tasks that matter to Healx in drug discovery for rare diseases. And of course, the most important decision that we have to make is, what disease are we going to work on? Clearly, if we select a disease that is more tractable, that has a higher likelihood of success in the clinic, then the whole process is likely to be more successful. Of course, it stands to reason. Working with our expert drug discovery scientists, clinicians, and pharmacologists, we've compiled a series of parameters, drivers of success in drug discovery, which we've built into an algorithm, a small little application, and we'll have a look at some of those properties in a moment. Effectively, we evaluate about 40 different parameters to determine whether a drug is, whether a disease is amenable to drug discovery. Having selected the disease to work on, of course, we're then to step 3. We're applying the AI workflows that enable us to do those 3 tasks I mentioned a moment ago. That is, predict monotherapies, predict combinations, having got the active candidate, work back to an understanding of how that drug might be working in the new disease. We call that process gaining therapeutic rationale, or indeed obtaining mechanistic insight. Of course, step 4, it's all about testing preclinically. It's about getting active candidates that we can either repurpose or develop as combinations. And you'll see down here that there's a little red circle reminding us that this is an AI learning loop. Okay, when we do our testing, our experimental testing, we want to input that information back into the knowledge graph so that we can constantly improve and learn in our algorithms. Really, really important point I want to make here is that our platform is a decision support tool. We're not trying to solve drug discovery with some kind of, black magic AI. We are indeed trying to help expert drug discovery scientists make better decisions more quickly. That's what's giving us the probability of success increase. It's the relationship between the AI platform and the humans who are ultimately making the decisions about which drugs we move further forward. And of course, this is a cloud-based platform, but I'm not the person to talk about the engineering aspect. I'm going to move further on then to what are the criteria that we're evaluating when we're understanding how to select disease to develop. There are 4 domains that we care about. We're evaluating what is known about the disease biology. We're understanding whether there are biomarkers and assays out there enabling you to undertake an in vitro and in vivo preclinical test. Can you test for efficacy? We're thinking immediately, right at the very start of the program, how are we going to run this in the clinic? Do we have access to clinical endpoints? Is there a patient group that we can partner with to gain key opinion leader networks? And of course, we are a company. There are commercial considerations around IP and freedom to operate, also market size access, all of those things we're considering. I'm not going to read through all of the labels on the screen down here, but effectively, these are top categories in our evaluation process for selecting diseases. There are some key ones. Not all parameters are equal. Clearly, having omics information that we can put into our drug-matching algorithms is quite important. Having those models is essential, as are knowing things like clinical endpoints and biomarkers for running a trial. Having selected a disease, we move on to the algorithms that are in the heart of our platform. On the screen now, we're looking at about 12 different algorithms that we've built at Healx that enable us to do that initial task in the central circle here of making a treatment prediction. Now, one of the first questions that we generally ask is, why do you need so many different drug matching algorithms? Why don't you just have a single algorithm that you apply that enables you to connect a drug and a disease? And it really comes down to the challenge of working in the rare disease space. 7,000 rare disease, 400 million patients. Actually, rare disease is relatively common. In the UK, it's estimated that about 1 in 17 people will be affected in some way with a rare disease. However, for any one of those rare diseases that we choose to work on, the availability of data, the coverage and the quality of that data can be patchy. Okay, so having multiple complementary algorithms that make connections in different ways using data and inputs in different ways allows us to have great access to the rare disease space, increasing that probability of success all the time. I'll talk you through the algorithm very briefly. In the top green box, algorithms known as new disease biology and mechanisms. These are algorithms that consume low-level molecular information. It's transcriptomic information, metabolomic information. So DGEM, for example, the first algorithm there, drug-gene expression matching, it's effectively using transcriptomic information, connectivity matching and money to find a connection between a drug and a disease. PASDA, pathway-specific drug analysis, kind of doing the same thing, but this time in functional space at the pathway level. Okay, drug reaction activity mapping, kind of like DGEM, but this time using metabolical reaction activity information. Actually uses a metabolic modeling system to predict reaction activities that we're using for MASHing. Those three algorithms, great if you have low-level molecular information as input. What if you don't? Okay, and in rare disease space, not surprising that gene expression information, high-quality gene expression information that is a good mimic of the disease biology is hard to come by. So then we have the algorithms in the bottom left-hand box, the orange box, knowledge graph and similarity matrices. These algorithms effectively need no more than the name of the disease to work on, provided that disease has an ontology and is in the graph. We can make a prediction between a drug and a disease. So DWPC, degree weighted path count. This is a meta-path exploration algorithm enumerating directly on top of the knowledge graph. It's kind of the Google Maps trying to plot a route between a disease drug node and a neighboring, a disease node and a neighboring drug node, and the kind of nodes and edges that you hop over can be an explanation for why that drug might be therapeutic in the disease. All well and good if there's no information in the graph that connects those edges. KBC, knowledge-based completion, perhaps our most exciting deep learning algorithm. Kind of important to us because it enables us to find true novelty, a connection between a drug and a disease, impute an edge, identify a link prediction for which there is no information supporting that connectivity within the graph. In some ways, it's kind of like the Netflix recommendation algorithm, and I understand going all the way back to transient, projection, embedding, not that different. Okay, PRISM, predicting indications with similarity matrices. This is using that kind of old-fashioned guilt by association concept in drug discovery. If you've got a drug and it's approved for an indication, then perhaps you can borrow that information to find a connection between a related drug and a related disease. That relatedness, in the case of PRISM, is obtained from log similarity matrices. Similarity matrices both in the drug space and in the disease space. And of course, there are 4,000 approved therapies out there that you can see that initial treatment relationship with. Both of these boxes predicting monotherapies. Okay, the first workflow that I introduced on the previous slide. What about predicting synergistic combinations? And it's important that we understand what we mean by synergistic combinations here. That's the idea that a pair of drugs given at lower doses individually can have greater efficacy than the sum of the parts. Okay, truly synergistic. And very important in the rare disease space where we're working with complex conditions with large symptomologies, where it's highly likely that a single drug to a single target is going to be truly therapeutic. So we've got 4 different algorithms that we've developed that ultimately predict synergistic combinations. COSINE, combination synergy evaluation. This is a fairly standard machine learning model built from experimental laboratory data. So you screen a whole bunch of pairs of drugs at various doses, determine whether they're synergistic or not, build a model, and then you can present that model with an unseen pair of drugs and make an estimation about whether that unseen pair of drugs is synergistic. Fine, you've got to keep that applicability domain when you're doing this, but that works quite well. And then we have network synergy. Network synergy, slightly different. Here, what we're doing is combining synergism, prediction, as in the first method, but also combining it with an understanding of the disease biology by building so-called disease modules using protein-protein interaction networks. And what we're trying to do here is replicate different aspects of a complex rare disease in different protein-protein interaction network disease modules and find synergistic pairs of drugs that target multiple modules. Okay. Finally, DGEM, combination gene expression matching, not surprisingly, very similar to DGEM, but this time we're using, rather than using our disease profile to query a drug signature database of single drugs, we've computed pairwise the gene expression profile of pairs of drugs. And we're trying then to find a computed pair of drugs that reverses that disease profile. Now, COSA, not really an AI ML algorithm. This stands for combination of single actives. And of course, if you've already made monotherapy predictions and you've shown them to be preclinical, it makes sense to simply pairwise test those pairs of drugs. And that's exactly what COSA does. So 3 boxes of algorithms that all serve the bottom task, making treatment predictions. What about evaluating or backtracking and understanding the mechanism? And here we've got algorithms called therapeutic rationale that do exactly this, generate hypotheses about how that prediction might be working in that new drug. Now, I won't talk about pathways enrichment, that simply represents all those standard computational biology methodologies. Of course, we do all those things, but I will talk extensively about an algorithm that we've developed called evidence chains. So what's an evidence chain? Well, this is an evidence chain, and it goes back to how our platform works. Remember, decision support. We've got a bunch of predictions, pharmacologists have to review them, they're picking the ones to move into preclinical testing, they need additional evidence to convince them that that's a worthwhile exercise. Of course, it gets quite expensive. The type of evidence that pharmacologists or drug discovery scientists and drug discovery scientists like to have is what is effectively a simple sentence that describes at the molecular level why that drug should work in the disease. So here's an example of the ideal sort of thing that pharmacologists like to have. I've got a disease, and that disease has some symptomology, some phenotype. That phenotype is underpinned by some disease gene, which impacts or perturbs a particular biological pathway. There is a drug, that drug binds to some target, and that target also operates in a shared or overlapping pathway to where the disease biology is coincident. If we can provide that information to the pharmacologists along with the predictions, that's great. And that's exactly what evidence chain does. It's effectively a way of querying the knowledge graph to find all of those evidence chains that has that kind of metapath structure based on compounds, phenotypes, genes, and diseases. And what we're able to do is effectively create a very small subgraph, providing all the known information about a particular prediction and how it relates to the disease. Importantly, we're labeling the edges with the impact or the relationship between the two nodes. So, for example, we might have a random prediction X01, and our graph tells us actually it's known to be in trial for epilepsy. Epilepsy is associated with mutations in UBE3A, and the UBE3A might be the disease gene that we're interested in. Moreover, here are a bunch of other drugs and how they relate to that particular disease. That information is quite informative in decision making. We'll have a little look in greater detail about how exactly we're using that information. So, how exactly is Healx then operating? I thought it quite useful to give an example of a particular program that we've undertaken. Our most advanced clinical program is in Pharmacological syndrome, and this kind of steps you through how we got to where we are today. First thing that we did is run our predictions in our platform. So the predictions, DGEM, PRISM, DRAM, KBC, all executed, made a bunch of predictions. All of those predictions have a metric that ranks the confidence that those predictions are going to be effective. And we take from each of those different methodologies the top around about 50 predictions, and we squeeze them all together and we present them to the pharmacologists with a bunch of standard annotations that they like to have through our drug rank discovery science tool, which I'll give an example of in a moment. And then we attach to those predictions, every single one of them, some kind of evidence chain that explains why we think it might be therapeutic. All of this, again, increases the probability of success of selecting something useful. Here's a little screenshot of the drug bank software. I'm not going to go through all of the columns. It's the usual sort of thing that you might expect to see. You know, just the Pharmacological, all the best predictions, great name for an experiment. And then in rows, all the compounds, got some little tags that may be proved or known to be in clinical trials at phase 4, so on and so forth. Mechanisms, yes, all that useful information. Right down on the last 2 columns here, direct and indirect evidence, kind of like I alluded to on the previous slide, presenting all of that information in a very digested format so that pharmacologists can make rapid decisions. I thought it'd be quite useful to drill down into what really an evidence chain is. And the first thing I want to do is not mislead anybody. Way back in 2017, when we ran the Healx study, we didn't have our fancy AI tool that I just described a moment ago. And we kind of did it the old-fashioned way. In fact, Dave Brown was a pharmacologist on the project. Dave, with 50 years experience in drug discovery, kind of a walking encyclopedia of pharmacology, took a look at a prediction, Healx M001, thought about the target genes, thought about the phenotypes and how they impacted Healx, and came up with a couple of articles that he kind of just happened to know. That is the second prediction made, Healx M001, although primarily a non-steroidal anti-inflammatory drug, is also PPAR gamma agonist, and the PPAR gamma agonists have been shown in Healx models of mouse models to improve cognition. Put those 2 pieces of evidence together and said, yes, we should test this particular drug. And that took several months for Dave to crunch through that information. These days, we have the evidence chains algorithm, and it can take a couple of hours to reconstruct that information automatically. You'll see down here that rank 11 of 7,000 evidence chains connecting Healx M01 with Healx. We have the explanation that Dave came up with. And we thought that's kind of good, cold justification. What we're really finding, though, is that most of our discovery scientists don't have 50 years of pharmacology experience, and therefore this tool can really enable scale of our process. I do want to just pause for a moment and say that, of course, going back to that original inspiration behind Healx, it was to address the larger 7,000 rare diseases. One thing I wanted to show on this particular slide by putting up our current Healx pipeline is that we've now run this platform on about 25 different programs, covering a wide range of therapeutic areas, neurodevelopment through to oncology and bone, and all of these drug discovery programs are now in progress. Of course, Healx is our most advanced program. We have IND approval. We're currently developing our clinical trial, but by the end of next year, it's our intention to also have clinical trials running for NF1 and ADPKD. They're kind of next in the pipeline. So let me just talk a little bit more about the Healx case study. For those not too familiar, Healx is a neurodevelopmental disorder. I've just listed the top symptoms, symptomologies over here. Got intellectual disability, hyperactivity, extremely sensitive to sound and light, sensory issues, autistic behaviors and seizures, a wide, complex range of symptomologies. In terms of monogenic disease, it's caused by repeat expansions in the FMR1 gene on the X chromosome, so clearly X-linked, meaning that boys are more affected than girls. We would argue that this is kind of a common rare disease in terms of prevalence, and obviously a large unmet need. Now, the gentleman you've seen over here is Mike Trasfaglia. And when I first met Mike back in 2017, to establish the partnership with FAXA, Pharmacological Association of America, Mike explained, he is a clinical psychologist and actually sees Healx patients. Mike and his wife Katie are also parents of 2 Healx kids. They established a foundation. It's a kind of a model patient group organization. Huge impact in raising awareness of Healx, generating funds for research. They also have their own drug validation initiative based in South America. and they've built mouse models of fragile X, meaning that they're able to rapidly test predictions. And back in 2017, we made 8 monotherapy predictions for fragile X. 3 of them proved to be effective. One of them, the most effective compound they'd ever seen. And that gave them confidence to work with us further to develop combination therapies. And a couple of years ago, we made 18 combination predictions. Half of them reversed 4 of the mouse behaviors completely, again, being incredibly effective. And clearly, it's those drugs that we're moving forward into trials. In fact, what we're going to do here is summarize the top 5 lead candidates that we've selected. And this is kind of also typical of the output from the platform. What it's showing is over on the left is effectively new disease biology, where in our hypothesis-free way, trying to develop novel targets for drug discovery, and you'll see those down here, a wide range of different targets. Namely the compound, the actual technology or the method that originally made that drug connectivity, and all of these have been tested in vivo. Not going to do well on this really complex picture of a nerve cell synapse, but what I just wanted to mention is that this is a figure from one of Liz Berry-Kravitz's publications, very respected clinician lead investigator on a number of fragile X clinical trials. In this publication, Berry-Kravitz wanted to summarize what are the common targets which are constantly evaluated from drug discovery in fragile X. These are the red boxes down here. GABA, mGlu5, ampakine. In fact, multiple trials have been run against mGlu5. It was the lead target for many, many years. All of which have failed. Of course, one of Healx's objectives was not to rediscover past issues, effectively to discover novel MOAs and also work in combination, because, of course, rare complex diseases, often a single drug is not going to cut it. The green targets down here are the ones that Healx has described working in very different biological pathways. So what are the kind of preclinical studies we carry out? I just wanted to give an indication of the sorts of studies that we performed for fragile X. There are 8 behavioral phenotypes in fragile X, which are routinely evaluated for efficacy, covering a wide range of human phenotypes. I'm just showing one over here, which is the open field test, which is a measure of hyperactivity, but there are others to do with social interaction, memory, and learning cognition. We've run all of those 8 panels against all of our lead candidates. But, what are we looking at on this particular slide? Well, over on the left, toward the y-axis, we have in the white box the wild-type mouse. Wild-type mice generally don't explore too far in an open field test, whereas the knockout, FMR1 knockout, fragile X mice run all over the place. And you can see here that we've given kind of 3 different doses of our lead drugs. In the first case, a limiting dose, a dose which is ineffective, and we get the same distance covered or the same hyperactivity measure as for the knockout mouse. And then we have a low active dose and a higher dose, both almost revert hyperactivity back to the wild type condition. Of course, again, 3 different lead drugs operating in PPAR gamma agonist, signal transduction modulator, and a GABA agonist. And one of the reasons why we believe that combinations are going to be effective is when we take those 8 behaviors that I've described a moment ago, aggregate them and summarize them, and look at really what the individual drugs are having an effect on in terms of human impact. And we can summarize human impact in terms of cognition, behavior, and anxiety. And we can see that, where we have a thick line, that drug is having greater impact. Where we have a thin line, it's having less of an impact. So, for example, the GABA agonist, really great on anxiety, less so on behavior and cognition. Clearly, if we only gave this as a monotherapy, only a small proportion of patients would respond, and that might have an impact on the statistical significance of the clinical trial that we run, where we have expected to get large amounts of heterogeneity. By giving combination therapies, we can cover much more evenly a broader aspect of the symptomologies of the disease. Where are we today? Well, we're just putting together our clinical trial. Not going to talk too much about this slide, just to say that lessons learned from others, we will be putting a placebo arm in here and having a placebo run-in, because it's usual in the cases of fragile X and for newer developmental and sort of clinical trials that patients automatically get a response irrespective of whatever drug they're given in that initial 12-week period, and we're going to try and counter for that effect. we'll be giving Healx 0201 or one of these drugs in low and high doses. We already know the dose that you give for Healx 0206. This was the gamma agonist. We've actually in-licensed this drug from its owners, Omic Pharmaceuticals, mainly. But the interesting story is, way back in 2017, when we made the original predictions, we actually predicted that this drug would be effective in Fragile X, but we didn't own the drug. Ovid have since tested that drug in a couple of other conditions and have withdrawn it due to its partial efficacy. We've now brought that drug in because we expect it will be synergistic with our other agents. Of course, by the end of around about next year, we shall have our initial readout, and that will determine whether we're going to progress with monotherapies or ideally a combination. Now, we might get lucky and find out that actually one of these particular drugs is effective more than we expect, and we will only need to progress with a monotherapy treatment. But the anticipation is that we will progress with a combination study. So I'm just going to finish off with a couple of slides more on the technology and the drug-matching algorithms that we've developed. And in the first case, I just want to remind you of the two drug discovery workflows we actually operate. Up to this point, we've really been talking about starting with a disease and trying to find treatment for that disease. Drug-matching, drug discovery. Clearly, that's what patient groups want. We start with a disease and we work with a drug. Our platform is also bi-directional, though. Our algorithms are able to operate in the reverse direction, where we can also start with a drug and perform indication expansion, identify a new disease that drug would be effective in. Particularly attractive to the pharma partners who might have a shelved asset and want to find a new indication for that drug. So these are the two workflows that our platform operates in. The first slide I'm looking at here, I appreciate this is quite a complex image on this slide. Let me tell you about it. Okay? What we want to do here is understand within those 7,000 rare diseases or disease at large, whether our drugs were equally effective in performing drug discovery, understanding the impact on therapeutic area selection for our platform. So what we did is set up a bit of a cross-validation exercise. We had, I don't know if you can see over here, 281 highly curated therapeutic relationships. That's a drug-disease pair. And we held that information out of our platform, covering a wide range of therapeutic areas. And we asked the algorithms find that held out information. And what you're seeing on the box plots at the bottom is the result of this study. Relatively complex, but I'll talk you through it. Along the x-axis, you have rank prediction, where if the algorithm predicts you rank 1, you're very confidently highly likely to be a hit. If you're rank 1,000, less confident. Okay? So you have rank on the x-axis, and up the y-axis, you have therapeutic area. Of course, we've had to aggregate those 281 drug disease relationships into top-level ontologies of therapeutic area. So we've got everything from autoimmune disease, respiratory cancer, all the way to cardiovascular. And the box plot that you're looking at, the length of that box plot gives you an indication of how effective that drug-matching algorithm is in making connections between a drug and a disease in that therapeutic area. So if you just stand back from the plots a little bit, what you're ideally wanting to see is everything squeezed up into the y-axis. That suggests the algorithms are working really, really well. The first observation that you see here, though, is that across the 4 different drug-matching algorithms, they're very different. The algorithms do behave differently in different therapeutic areas. However, if you squeeze them all together into a combined or aggregated prediction methodology, they all work really, really well, okay, as a combination. Again, more evidence for the reason why you want multiple drug-matching technologies more than one. And of course, this is not really a novel concept in AI or machine learning, clearly ensemble models of the products of multiple models together. And that's really what we're doing in the technology platform. So we've got an understanding of how the methodologies work in different therapeutic areas. What about the individual methodologies themselves? So I've just got 2 examples, one in DGEM and one in KBC. And just a reminder of what drug gene expression matching really is. It's connectivity mapping in old money. You have a disease gene expression profile that you're going to use as a query signature, and you're going to use that to query a database of drug signatures. Ideally, you're looking for a drug signature that induces an equal and opposite effect to the disease, because that's the issue aversion. The idea is that you're going to compensate the effect of the perturbations in the disease, result back to balance restored or normality. Not really an AI algorithm, it's actually more of a statistical methodology. And really what you're getting out of this is a connectivity measure. That connectivity measure is going to rank your drugs between those drugs which are most dissimilar to your disease gene expression profile, that would be inverse connectivity matching, all the way through to correlated positive connectivity. So your drugs in your library are then ranged from 1 to, in this case, about 1,000, given that little blue squiggly line down here. So if you're on the end of the here, you're very negatively connected. If you're on the end on the other side, you're very positively connected. And of course, that little DGEM algorithm spews out its information straight into the drug bank software with all the annotations that pharmacologists can review. Doesn't tell us how well it's working. Doesn't say how good that connectivity is. Is that drug over there on the y-axis strongly connected? Are we very confident, or is it not? We need a measure of performance here. How strong or confident are we in that connectivity? And we came up with the little idea of we can seed in the ground truth drugs and use those as an enrichment calculation to determine whether the connectivity is meaningful or not. It just so happens that this is real world data. This is acute myeloid leukemia here, and there are 16 ground truth drugs for acute myeloid leukemia in our signature database, and we can measure their enrichment to know whether this is good or bad connectivity. And that's what you're looking at on this slide here. So I've just zoomed in the ground truth drugs. And you might just see a little bit of a pattern for me here. We thought this was quite surprising. Over here, you've got a clump of kind of negatively connected ground truth drugs. And over here, you've got a clump of positively connected or correlated drugs. How on earth are these drugs which have the same profile as the disease having therapeutic benefit? One to think about. Moreover, though, what does connectivity matching actually look like at the gene level? And what you're seeing down here are the gene readouts. These are the genes that are actually driving the connectivity between the drug and the disease. And what I've done is I'm just pointing out the most negatively connected drug, which is tretinoin or retinoic acid. And over here, you've got on the positively correlated side, the most extreme one was doxorubicin. And you can see that in the disease gene profile, I'm hoping I can convince you that where the genes are downregulated or red in the disease, we'll find out whether green or upregulated in the drug, and where they're up in the drug, green, and down, red in the drug. That kind of visual indication of what connectivity mapping really looks like, but how can we use this again for knowing whether this is good or bad connectivity? And that's where this little precision decay plot comes in. What we've done is we've taken the 50 most extreme positive, the 50 most extreme negatively connected drugs, absolute connectivity, ordered them along the x-axis down here, and then just simply counted up the number of ground truth drugs that we found. So if we go straight to rank 10, we're finding or rediscovering 2 of the known treatments for AML. A bit of an enrichment analysis, that's 18 times greater than random chance, or you expect to see that 1 in 5,000 experiments. So we feel that's quite confident way of knowing that the connectivity is actually working. Okay? More importantly, the 8 drugs higher ranked than the 2 control compounds might actually be interesting drugs to look at. Of course, in this kind of drug discovery research, positive hits, great, no problem, but you don't know that something is truly a negative. It simply might not have been tested. Okay. So that's all well and good for statistical methods of connectivity. What about machine learning methods, AI approaches? So let's have a look at how we do a similar sort of thing for knowledge-based completion algorithm. Again, remembering that there were 2 workflows that we're looking at here, both drug discovery and indication expansion. And the 3 questions that we're really trying to answer on the slide here. Can our models predict novel, unseen connectivity, known relationships? How accurate is it in doing it? And do we actually know that the algorithm is learning treatment relationship? Is there actually learning taking place? And the way that we do this is by training our knowledge graph, or building our knowledge graph and training the models up to all data up to November 21, and holding out all new discoveries ahead of that time. There are 507 novel indications between the end of November 21 and about 6 months ago. Okay. Can we rediscover them? It's that straightforward. The first thing that we do, though, is try and get a bit of a baseline. Again, I mentioned earlier on, or by chance, that the way that we build the knowledge graph and by projection embedding, okay, which basically produces a large similarity matrices, okay? And we can actually use that matrices that underpins the knowledge graph to get by association style connectivity. We're going to use that as our baseline. So basically not turning on the learning. Yeah. So what we're looking at down here then is the two separate methodologies. We've got indication expansion task. We've got the drug redevelopment or drug discovery task. Along the x-axis, you've got hits at or top rank predictions, hits at K. And up the y-axis, you've got percentage recall. How many of those 507 held out the drug can we discover? And yay, not doing too badly. If we take the top 100 ranks, we're getting for indication expansion about a fifth of those held out indications, connections rediscovered. For drug redevelopment, we're getting around 40%. Okay. Why 100? Turns out that 100 is the maximum number of predictions you can give to a pharmacologist before they hit you in terms of evaluating them. Okay. Worthwhile knowledge. Important to know, in fact. Okay. So what happens when you turn on the learning? Wow. For indication expansion, you get about a fourfold improvement in discovering those held out 507 indications. For redevelopment, we get about a twofold improvement. That's pretty dramatic, actually, and actually makes a difference. It answers the first and second question: can we discover novelty at all? And can we measure it? But the third one, I think, is quite tricky. Do we know that it's learning? That's kind of hard to unpack. What we're taking here is look at the shape of the curve to the KBC line. That hump towards the y-axis tells us there is an enrichment of truth at the top end of predictions, the predictions we care about the most. This is really fundamental for probability of success and scaling your process, because it kind of means that pharmacologists don't need to review 100 predictions. You might be able to give them 50, and they would have the same hit rate. So we felt that that's a good indication that we are doing some learning and that it is having an impact on the way that our platform works. So where do we go? What comes after this? Again, here's a picture of Dave Brown, our co-founder. Some might know him as the former global head of drug discovery at Roche, but he's probably more famous for the fact that he's the guy that was co-inventor of Viagra when he was at Pfizer. Dave and Tim listened to Nick speak right at the start, wanted to understand how we could scale this process to address all medical unmet need in their disease space. Could we build something which is more autonomous and scalable, that combined AI drug discovery with also wet lab capabilities to rapidly evaluate and test those processes, feed that information back into the learning and improve over time? That's what we're currently building and developing at Healx, and that's what's next for us. Now, I'm just going to finish by putting this slide up just to say that we are interested in hearing about your discovery experiences and stories, thinking about possibilities for collaboration and partnership. There are kind of three areas that we are looking to partner in. One is around that discovery and disease biology. One is around indication expansion, and also potentially adding a second agent to a known active to increase efficacy, companion or combination discovery. Thank you very much for listening. I'm more than happy to answer any questions.